to thank Jay and Wolfram and the NIH and the DZHK for organizing, and all the technical and administrative staff for making this session and this conference so wonderful. So I've been asked to talk about regulation of heart rate and rhythm by CamKII. It's a subject I love. And to do that, I'm going to just introduce a topic in a slightly more detail than was already brought up, which is this concept of clocks. And here I'm referring to work to help to understand automaticity in cardiac pacemaker cells per se, but I think that broadly speaking, these concepts are true for mechanically purposed cells, especially when they become arrhythmogenic. So the calcium clock is pictured here in this, there. And really what it is a sarcoplasmic reticulum calcium recycling apparatus that is present for EC coupling in mechanically purposed cells, but which lives on a percentage-wise basis just as large in these pacemaker cells that are not mechanically purposed. And it's a clock because even when you uncouple it from the membrane clock, it will go for a while all on its own. The membrane clock in the pacemaker cell has very specific components. We heard about one of them, the so-called pacemaker channel, mostly HCN4, but it has a number of other calcium-sensitive conductances, notably the sodium-calcium exchanger, and then one type of voltage-gated calcium channel, the L-type calcium channel. The details of these membrane conductances, of course, varies across development and across different mechanically and pacemaking purpose cells, but the general concept holds. And again, mostly because of the HCN4 channel, this automaticity will happen even when this is turned off, at least to a certain extent. Another way of displaying these concepts is through the action potential, which shows this regenerative reactivation of action potentials, and that really is due to this, sometimes people call it phase 4, this late diastolic event where the membrane potential is slowly depolarizing. And when it does that, it's in the presence of a kind of a foot of calcium release. When it reaches a critical potential, mostly in the central pacemaker cells, voltage-gated L-type calcium channels open up and make for this action potential steep rising phase and contribute to a trigger that leads to more calcium release from the sarcoplasmic reticulum. And so these two concepts live together and can be coupled by various signals. And one of them is this molecule, CamKII, which I'm going to talk about. And then I just think these concepts are broadly useful in thinking about the automaticity that we talked about in Dr. Laflamme's talk, and that I think helps to understand automatic rhythms in failing hearts. So there's a cartoon of CamKII. CamKII is the multifunctional calcium and calmodulin dependent protein kinase 2. It's a serine threonine kinase. It's richly abundant in striated muscle, immune cells, a few other cells, but notably here in heart muscle and including in pacemaker cells. So this cartoon shows imagines a monomer of CamKII. The CamKII holoenzyme is a dodecamer or a 14-mer that consists of 2 stacked plates. Under resting conditions, CamKII is mostly inactive, and that's because, like many other kinases and phosphatases, it has an autoinhibitory sequence that acts as kind of a decoy that lies over this activation site, and it constrains the kinase, making the ATP binding pocket unavailable. When calcium rises in the cell, and in pacemaker cells it's rising rhythmically, and in heart cells it's rising with every contraction, calcium complexes with this calcium-sensing protein calmodulin, which is ubiquitous, and calcified calmodulin binds in a very specific region of the CamKII regulatory domain, causing a steric change that removes the autoinhibitory sequence from this active site, causing CamKII to be open. Now this can go back and forth. Calcium rises and falls, CamKII or calmodulin becomes calcified and then decalcified, so the kinase becomes active and inactive. except that there's another layer of regulation through post-translational modifications. And I just listed a few of them here. All of them kind of do the same thing. They change the charge structure of these amino acids so that they're no longer good at sitting in this very hydrophobic spot and leave the kinase in a calcium-independent state of activation. The ones I'll point out is the canonical one, that this autophosphorylation site, this threonine 287, I'm going to show you some antibody work that may reflect activation of CamKII here. A site that activated by oxidation of paramethionines that our group helped to identify about 10 years ago. And then a serine right here that may be activated by oglycanacylation, a metabolite of glucose. So this is an example of some rather old work. It was led by Yujin Wu, who is technically a master, and it followed some work we did early on when we made some CamKII inhibitor hearts, just using the alpha-myosin heavy chain promoter to express a CamKII inhibitory peptide that's actually based on, modeled on that regulatory autoinhibitory sequence I walked you through. Fused it with green fluorescent protein. You can see these green hearts. We also made a scrambled version of that peptide, AC3C for control, that doesn't have any inhibitory activation. And although this was not the focus of this paper, we noticed this very decisive decrease in heart rate in the CamKII inhibited compared to the transgenic or the wild type control. And so a number of years later, Yujin Wu came to me and said, I think I figured out how to isolate these pacemaker cells. So, for people in the grown-up myocyte business, these hardly seem like cells at all. They're very long and stringy. There's actually quite a diversity of their phenotypes, and they quiver, but they are clearly not mechanically purposed. And here you can see, so this is wild type, AC3C and AC3I under baseline conditions, and then when we give isoproterenol. And so these are all green because they express this transgenic protein. But after isoproterenol, you can detect this activated form of CamKII throughout the cell, except in the AC3I cells, which is what the model is supposed to do, and led us to make further measurements, this one in collaboration with Longshan Song at Iowa. But what you can see here is this is an overlay tracing of spontaneous calcium events in the pacemaker cell and the action potential in red. And when you get down to look at the superimposition of two of these action potentials, and remember, the action potential is influenced by the calcium, and the calcium influences the action potential. So there's this coupling that we see that the steepness of that phase 4 is enhanced, and that's from the blue to this rather faint orange-red. It's enhanced by isoproterenol. Isoproterenol increases your heart rate. And that's true in the control transgenic, the wild type. But with the CamKII inhibited one, that change doesn't happen, and isoproterenol is much less potent at increasing rate. So this and other kinds of evidence led us to conclude the CamKII was important for about 50% of the dynamic range of heart rate increases in the face of catecholamines. So CamKII seems to be important for the fundamental physiologic signaling of heart rate. This is work that a fellow, now a faculty at LSU, did in the lab some time ago when he infused mice with angiotensin II over time and noticed that these mice had a sick sinus-like phenotype. This is a phenotype where the heart rate would get very, very slow, and then it might come back a little faster. It was quite irregular. And he ended up mapping that to an oxidized form of CamKII. So here's some data in pacemaker cells stained for the funny current that's carried through this HCN4 channel, which is enriched in the pacemaker region, and then a stain for oxidized CamKII after angiotensin II, which in other work we and others have shown activates CamKII mostly through an oxidative mechanism compared to saline, where we didn't see that. And in patients in a model of canine heart failure, we found something similar. So this is total CamKII, this oxidized CamKII from patients where hearts that didn't require a pacemaker, patients with heart failure that didn't require a pacemaker, and patients with heart failure who did require a pacemaker, suggesting that CamKII might be important for the physiology of pacemaker behavior, but also it's bad for pacemaker cells, and it causes a kind of cell death, data I don't have to show you, can't show you here, and that when a critical number of pacemaker cells die, it alters the source-sink relationship between the small group of pacemaker cells that routinely are injecting current into the larger myocardium. And as they become less efficient, it creates this pacemaker phenotype. And in fact, one of the most sort of stalwart bioresponses is with aging, when your upper heart rate is limited. There's a sort of out-of-your-pocket calculation that works pretty well, and at least in rats, we see a similar age-related decline in the mass of the sinoatrial node, the pacemaker collection. We find something similar happens in a model of diabetic cardiomyopathy, where diabetic mice after myocardial infarction die in greater numbers, and we map that to an augmented pathway that looks very similar to what I showed you schematically with angiotensin infusion, that there's more sinus node dysfunction from excessive apoptosis leading to low cardiac outputs and mortality, and that could be reversed through CamKII inhibition. So, but I started talking about CamKII doing things to the living cell without killing it, and I just want to touch on that just for a bit. So these are not pacemaker cells, these are atrial cardiomyocytes, isolated from mice that were infused with saline or angiotensin II. Remember, angiotensin II activates CamKII, we think mostly through this oxidative pathway. And here, we take wild-type mice or knock-in mice where the methionine pair, remember, that activates CamKII through oxidation is replaced with valines, which can't be oxidized. So with saline, and these little starry skies are just really fluorescent reporters showing calcium release, mostly from the sarcoplasmic reticulum. And what you can see is that increases with angiotensin II and wild-type, but not so much in these mice where everything is the same, except they don't have CamKII that can be activated through oxidation. When we look at that in summary, you can see that very clearly, that you need that methionine pair, the ability to oxidize CamKII for activation to respond to angiotensin. So maybe nobody cares about that, but lots of people care about atrial fibrillation. It's very common, and it's particularly common, it turns out, in diabetes, which are both of these, as we've already heard in many talks, diabetes is a huge challenge in cardiovascular disease. So is atrial fibrillation. It turns out that diabetes is an independent risk factor in many studies for atrial fibrillation. And if we make a mouse diabetic, we can do it in a couple of ways with a type 1 model or type 2 model and pace the mouse. We can readily induce atrial fibrillation. This is done by Rotimi Musubi, who's now an assistant professor at Hopkins. And what he found is that diabetes increases the incidence of atrial fibrillation, but if you inhibit CamKII because you express that transgenic protein AC3I, or you're a knock-in mouse that can't have oxidized CamKII, or maybe if you lack that oglycanacylation site, oglycanacylation is more robust with diabetes, that you're protected. We also found that there's a modest cardiomyopathy in type 1 diabetes, and that mice with this, with the MMVV that are resistant to oxidation are also resistant to that mild ventricular dysfunction. I just want to touch briefly on, so CamKII is kind of I think is a kind of a supercharging molecule. It can be knocked out, it's pretty dispensable. But when we were looking at its post-translational modification, Sinchuan Wang, together with Gab Bever, who's an evolutionary biologist, noted that these methionines really are an invention of vertebrates. They don't happen in invertebrates. CamKII is about 1 billion years old, and this is a new invention. So why is it? Well, we found that mice that lacked CamKII that could be oxidized through knock-in mutation weren't as fast, and they couldn't run as far. And so we wondered if this oxidation occurred in vertebrates so it could supercharge metabolic responses for fight or flight. And to look at that, we looked at the fly, which is much simpler. It has one CamKII, and it has valines rather than methionines. And when we made a knock-in mutation for those flies and looked at their heart tubes, well, we also found they could climb faster. But their heart tubes are hyperdynamic. So they're kind of like superflies. But if we look at the echocardiogram of the heart tube, let's see if I can do. What I think you can see is that at baseline you can see very clearly the wild type hearts don't really suffer when you treat them with paraquat, which is an herbicide that makes oxidant stress through a variety of pathways. So you can see that here and here. But if you look at these these humanized or vertebrate flies, they start out just fine. In fact, they have super hearts. I think I didn't get that one going. But what you can see is that they develop arrhythmias. We declared them as moderate, severe, or asystolic. And you can see the summary data here. So adding those methionines created not only a super physiologic performance, but a heightened susceptibility to disease stress related to oxidation. So I want to conclude just to say that pacemaker cells are governed by a membrane clock and a calcium clock. We're going to probably hear more about that, at least in other contexts. That CamKII plays a physiologic role to increase heart rate, in part by enhancing calcium release from ryanodine receptors, and that too much CamKII can kill pacemaker cells and other cells, and contributing to a sick sinus syndrome type phenotype. The too much CamKII, CamKII is probably a supercharging molecule, but too much of it is bad, and it helps to conceptually explain, at least in my view, the connection clinically between heart failure and arrhythmias. And finally, the potential to activate CamKII through oxidation was acquired at a very clear juncture at the very beginning of vertebrate biology with hagfish and the kind of animals you learned about in high school. And that provides this enhanced physiologic performance in mice and in flies, but also enhances the susceptibility to cardiac diseases. I just want to list a lot of the people who have helped with this and related findings, and to thank you for your attention.